Welcome to Investigative Biology. This week, our scientists go beneath the surface and uncover HIV. Good afternoon. I'm Colin Weaver, your host for Investigative Biology. Since its discovery in 1981, the human immunodeficiency virus, or HIV, has killed more than 25 million people worldwide. In 2008 alone, the World Health Organization estimated that there were 33 million people living with HIV, in addition to 2 million deaths and 2.7 million new infections. Known in its later, more fatal stages as the acquired immunodeficiency syndrome, or AIDS, HIV is perhaps the most notorious killer of our age. It is spread through unprotected sexual intercourse, breast milk transmission from mother to child, and other blood exchanges such as contaminated needles or blood transfusion. Whereas a bacterium is a one-celled living organism defined by its ability to reproduce, illustrated here. A virus is a mere protein coating surrounding a DNA or RNA molecule.
I need you.
Infection begins when the virus attacks the immune system's lymphocytes, particularly the CD4 cells. But more on that later. For now, let's head out to our scientist in the field, Jonny Reindollar, for an overview of the viral structure.
1/100th the size of a TB bacterium. The human immunodeficiency virus contains an outer viral envelope. Directly attached to this outer viral envelope is the glycoprotein 41. And directly attached to this glycoprotein 41 is glycoprotein 120. The glycoproteins rotate spherically around the virus, like so. The viral core, which is spherical as well, contains 2 single-strand molecules of RNA, as well as the protein enzymes reverse transcriptase and integrase. Here is a simplistic diagram of the viral core. Please take note, 2 single-strand RNA molecules, the enzyme reverse transcriptase, and the other enzyme integrase.
Now that we have a better understanding of the viral structure, you may be wondering how that structure lends itself to killing CD4 cells. But first, we must have a better understanding of why CD4 cells are so important to the immune system. Here is our own animal reenactment. Starring Bob as a TB bacterium, Genevieve as the immune system's macrophage, myself as a CD4 cell, complete with an antigen marker to identify foreign invaders, and Jonny as HIV.
The CD4 cell is strolling through the bloodstream. Oh look, it has found a TB bacterium, and it has marked this TB bacterium with its antigen. The antigen is used so that a macrophage can identify a bacterium as a foreign invader. And thus, here comes the macrophage to eat up this bacterium. Look, it found the antigen. Bye-bye, bacterium. This is how HIV affects the immune system. Now the CD4 cell is unable to mark the bacterium with its antigen. And alas, the macrophage can no longer identify where the bacterium is. CD4 cells are very important to the immune system, and without them, co-infection is likely to occur.
Now that our ignorance of how CD4 cells work has been squashed out, we return to Jonny Reindollar.
When the HIV virion bumps into a CD4 cell, the outer proteins, glycoprotein 120, actually bonds with the receptor proteins on the outer envelope of the CD4 cell, forming this lock and key combo. Now, when this lock and key combo occurs, a hole actually develops in the CD4 cell, called the fusion core, and the inner viral core actually injects itself through the fusion core into the host CD4 cell's cytoplasm. Now in the cytoplasm of the CD4 cell, there are 2 RNA strands from the viral core, as well as the enzymes reverse transcriptase and integrase. What happens here actually is the enzyme reverse transcriptase binds itself to an RNA molecule and helps pair off nitrogen bases to form a DNA molecule. The same occurs for the other RNA strand as well, forming, again, another DNA double helix molecule. Together, these DNA molecules and proteins make their way through a nuclear pore and into the nucleus. In the nucleus, the DNA molecules, with the help of the enzyme integrase, actually integrate themselves into the CD4's DNA. Once the viral DNA is now integrated into the CD4's DNA, a DNA transcriptase enzyme forms this RNA molecule that actually heads straight to a tiny organelle called the ribosome. At the ribosome, a messenger RNA molecule forms itself into a protein. So when this messenger RNA makes its way through the ribosome, several proteins can be printed out, such as glycoprotein 41, glycoprotein 120, the enzyme reverse transcriptase, or the enzyme integrase. Now, once you have all this congregation of these proteins at the cell's surface, another strand, or 2 strands of messenger RNA can actually make their way to that exact congregation, and together they can all butt off into more HIV viruses, ready to infect other CD4 cells, weakening the immune system. Treatment includes antiretroviral therapy, which delivers reverse transcriptase inhibitors to prevent the viral DNA from forming in the first place. A separate treatment involves protease translational inhibitors, which prevent messenger RNA from forming the viral proteins. Both solutions prevent the creation of further HIV viruses.
That's it for this week of Investigative Biology. Please join us next time as we continue to wash away biological mysteries.